Sushruta for NEET is now available on the Google Play Store. Try it out for free.
We were studying about genetically modified organisms under biotechnology and applications. These genetically modified organisms mainly include plants, bacteria, fungi, and animals whose genes have been altered by manipulation for our requirement. This genetic modification has made crops more tolerant to abiotic stresses. Abiotic stresses are stresses due to conditions like extreme cold, drought, salt, heat, etc., etc. And also reduced reliance on chemical pesticides. Now, for each and every crop, we have to use one or the other chemical as a pesticide. Pesticide kills the pests. Pests are unwanted creatures which are harming our crops. So it also helps to reduce post-harvest losses. Many times, after harvesting is done, say tomato is grown and it is readily ripened, we have cut them. But prior to marketing, since chances are there that all of a sudden the rate comes down, farmers will not come forward to sell at the low cost. Sometimes there will not be demand at all. And this results in post-harvest losses. GMO also increases efficiency of mineral usage by plants. This prevents early exhaustion of fertility soil. These genetically modified organisms help us in a different way. As I have already told you, many times we use these inorganic fertilizers so much so, the soil loses its fertility. In the sense, without adding this inorganic fertilizer, you cannot grow any crop. So, as a result of which, what we do? We put more and more fertilizers to get more and more yield. Too much usage of these inorganic fertilizers are also not good to the soil. So increased efficiency of mineral usage by plants should be there. With less input, we should get good output. And also, this GMO has enhanced the nutrition value of food. For example, vitamin A enriched rice. We shall see all these, how it is possible to get in by studying individual cases. In addition to these uses, GM has been used to create tailor-made plants to supply alternative resources to industries in the form of starches, fuels, pharmaceuticals, etc. Okay, let us look into some of the examples. Some of the applications of biotechnology, especially in the field of agriculture, that we are going to study in detail for the production of pest-resistant plant. Even if at all some insect tries to attack that plant, plants will develop resistance power to such an attack. So, applications of biotechnology in the field of agriculture has resulted in the production of pest-resistant plants, which could decrease the amount of pesticide used. We need not to go for our traditional use of pesticides because of the some quality that the plant has developed, what is called pest resistance. For example, Bt toxin is produced by a bacterium called Bacillus thuringiensis, therefore short form Bt. You might have heard about Bt cotton several times. Bt here, B refers to the bacterial genus, thuringiensis refers to T, refers to the species. So it is a Bt in short. Always ask a question, what is Bt? This is the bacterium Bacillus thuringiensis. Now let us see what is the speciality of this Bacillus thuringiensis. In what way this Bt, Bacillus thuringiensis, is used in improving our agriculture? Bt toxin, this toxin got developed by the bacterium Bacillus thuringiensis. Bt toxin gene has been cloned from the bacteria. We have removed that gene from the bacteria and been expressed in plants to provide resistance to insects without the need for insecticide. In effect, created a biopesticide. That means biopesticide is a pesticide where biological weapon is used to get rid of the disease-causing insects. Biopesticide. Here, living organism is used as a pesticide in spite of the or instead of the chemical. Let me repeat again. In the Bt toxin, that means the toxin which we get it because of the Bacillus thuringiensis gene. Bt toxin gene has been cloned from the bacteria Bacillus thuringiensis, and this gene has been introduced into the plants of our choice to develop a resistance to insects. So once this Bt toxin gene is introduced into the crop in which we are interested, that crop will not be affected by the insects. So you need not to use the insecticide which we are using before to get rid of that insect. Examples are Bt cotton, Bt corn, rice, tomato, potato, and soybean. In all these, we have successfully introduced Bt toxin gene to see that they develop resistance to insects. Have you followed this? Have you followed this? Pratiraj,
Yes, sir.
number one,
Yes, sir.
Diti,
Yes, sir.
Savitri,
Yes, sir.
Tanusri,
Yes.
Jayasri,
Yes, sir.
Nadira, Vispita, Kanisa, Navya, Blessy. What? Are you not hearing me? Navya?
Yes, sir.
I wanted to make it sure that you people are there. That's why I am taking attendance in a different way. Are you there? Sleeping? Navya?
No,
Lohit?
sir.
Yes, sir.
Eating? Lohit?
Yes, sir.
Okay. Harshvardhan?
Yes, sir.
Siddesh? Charan? Are you there?
Yes, sir.
Harshvardhan?
Yes,
You
sir.
are all there. You?
What, sir?
Who are all there with you there?
Sir, Charan.
Then Siddesh is
Sarvatam.
there. Sarvatam is there. Then,
Siddesh, I don't know, sir. Sarvatam is there.
okay, who else? You are only 3 members from hostel.
No, sir. Vishwas also is there.
Vishwas is there. Has he come to the class?
Sir, his phone switch off, sir. So
Huh?
is we.
His phone is not working.
No, sir.
Your
Sir,
phone is working.
my phone is switched off, sir. That's why I am listening in Harsha's phone.
Okay. But I should know, that you are all there.
Yes, sir. We are all there, sir.
Okay. Right. So, we shall continue now. I hope you have understood this. Sarvotam, have you
Yes,
followed
sir.
this? What is
Yes,
done
sir.
with the BT toxin? What is done with the BT? Bacillus thuringiensis? BT toxin producing gene is introduced into some of our crop plants to develop resistance to insects so that we need not use much of these chemical pesticides. Understood? Clear? Look here. Now we shall look into BT cotton. It will be asked in the examination. Some strains of Bacillus thuringiensis produce proteins that kill certain insects such as lepidopterans, like a tobacco button, armyworm, coleopterans, like beetles, and dipterans, like flies and mosquito. What is that? Some strains of Bacillus thuringiensis, this bacteria, some strain in bacteria we do come across different strains. Some strains of Bacillus thuringiensis produce proteins, and these proteins kill certain insects such as lepidopterans, coleopterans, and dipterans. Bacillus thuringiensis, what it forms? It forms protein crystals during a particular phase of their growth. At a particular phase of their growth. In a sense, when you study these insects, first they pass through their larval stage called a caterpillar larva. So, this particular, whatever that proteins we add, these proteins produced by this Bacillus thuringiensis, either they kill these insects in the larval form or in the adult form. So, Bacillus thuringiensis forms protein crystals during a particular phase of their growth. Not that throughout the life cycle of the insect. During a particular phase of the life cycle, you will see that. Bacillus thuringiensis forms protein crystals during a particular phase of their growth. These crystals contain a toxic insecticidal protein. Whatever the protein crystal is there, it contains insecticide. Why does this toxin not kill the bacillus? That is the question you may ask yourself. If at all Bacillus thuringiensis produces a protein, and if at all it is going to kill the insect, why this protein is not going to kill the Bacillus thuringiensis itself? This is the question. Now let us see how it is produced, when it will be effective. Please listen to me. We are now speaking about a protein produced by Bacillus thuringiensis, and this protein is going to kill the insect which is harmful to us, like lepidopterans, coleopterans, or dipterans. I shall show you this. These are the Bacillus thuringiensis, and these are extensively used in BT cotton. That is the cotton which has developed a Bacillus thuringiensis gene to have the insect resistance. Lepidopterans, order Lepidoptera. You must know what are these order Lepidopterans, what are the coleopterans, what are the dipterans. Order Lepidoptera includes butterflies and moths. This is what the Lepidoptera. You must know Lepidoptera means butterflies and moths. Coleoptera means beetles. Dipteran means flies, mosquito. Your house fly like that. All flies comes there. Your cockroach also comes under the dipterans. So lepidopterans, coleopterans, and dipterans. These are treated with the genes or the genes developed by the Bacillus thuringiensis, which produces certain protein, and these proteins they harm these insects in a particular stage of development. So this is tobacco budworm, which is going to spoil the tobacco bud's leaves. And this is armyworm. This is whatever beetles. This is now actually the Bt toxin protein exists as inactive protoxins. Protoxin. Always pro, whenever it is added to a word as a prefix, it is inactive form. So the Bt toxin protein exists as inactive protoxins, but once an insect ingests the inactive toxin, once the insect ingests inactive toxin, it is converted into an active form of toxin due to the alkaline pH of the gut, which solubilizes the crystal. I repeat again, when an insect, it eats its protein, at that time, when the protein enters into the body of the insect, it will be an inactive condition. It is called inactive protoxin. The Bt toxin, that is the toxin developed by the Bacillus thuringiensis bacteria, the Bt toxin protein exists as an inactive form, that is protoxin. But once the protoxin enters into the insect gut, in the gut, alimentary canal of this insect, it gets activated because of the alkaline pH of the gut. So now it becomes active. That means whenever it is in inactive form, it was not poisonous. When it gets activated, it becomes poisonous. The activated toxin binds to the surface of the midgut epithelial cell. It binds to the surface of midgut epithelial cells and create pores in the gut of this insect that causes cell swelling and lysis and eventually cause death of the insect. So whenever the insect goes and sits and tries to eat or damage its crops, immediately this Bt toxin enters into the body of the insect, but it is there in an inactive condition, inactive protoxin. So because it is an inactive protoxin condition, this toxin is not going to harm the bacteria itself. So only this inactive protoxin, when it is introduced into the gut of the insect, when it is introduced into the alimentary canal of the insect, when this toxin comes to the intestine, the gut, say alimentary canal, foregut, midgut, hindgut, whenever it comes to the gut, in the gut, alimentary canal of the insect, since there is alkaline medium, and this alkaline medium solubilizes the crystal, and the toxin gets released, and it makes the midgut epithelial cells to act upon or to get damaged, and pores get formed there, and this creates the swelling of that particular part, and lysis takes place, eventually causes the death of the insect. Did you understand this concept? Did you understand, Janani?
Yes, sir. Yes, sir.
Have you followed Harshavardhan, Sarvottam, Savitri,
Yes, sir.
Kavita? Kavita, have you followed?
Yes, sir.
Okay. Shall I go ahead, Kanisa?
Sir, she's having network
Kanisa?
issue.
Pardon?
She's having network issue, sir.
Who has network issue?
Kenisa.
Who is speaking now?
Janani.
Janani. Okay. Janani, whether Aisha has come? Okay.
No,
Now,
sir.
Janan, yes. If now you understood, let me go ahead. This is what? Cotton ball. One, here, A, destroyed bollworm. Once the bollworm makes a hole like that, then the cotton threads, whatever you come across inside, it cannot be used at all to manufacture the fabrics. A fully mature cotton ball. Specific Bt toxin genes were isolated from Bacillus thuringiensis, specific for a specific crop, and incorporated into the several crop plants such as cotton. Now let us see some of these different beta toxin genes. The choice of genes depends upon the crop and the targeted pest, as most beta toxins are insect group specific. Most of the beta toxins are specifically used for insect pests. Okay, the toxin is coded by a gene named CRY. So CRY gene is actually concerned with the production of the toxin, which biotoxin, which it always acts on the insect group. There are a number of them. CRY genes, for example, the proteins encoded by the genes CRY1AC, CRY2AB control cotton bollworms. Once these CRY1AC, CRY2AB proteins are introduced or gene producing these proteins are introduced, genes CRY1AC, CRY2AB are introduced into this cotton. This cotton develops a resistance to the insect and cotton bollworm cannot grow there. It will not survive there. Okay, so no more it can affect this plant. Like that, you have K1AB controls corn borer. Okay, you must know these different genes which are used as Bt toxins. So you just know Cry2AB, Cry1AC, they are specifically used to control cotton bollworm, Cry1AB to control corn borer, Cry3AB to control Colorado potato beetles, and Cry3BB, it is for corn root worm. So you can have this Cry2AB, Cry1AC for cotton bollworm, Cry1AB for corn borer, Cry3AB for Colorado potato beetle, and Cry3BB to control corn root worm. Clear? Understood? In this way, we have developed different genes to treat different types of insects. Right? Clear? Clear? So these are all produced by using Bacillus thuringiensis strain. Have you followed? Are you following me? Clear? Jyoti?
Yes.
Namrata?
Yes, sir.
Okay. This is toxin Cry1AC from Bacillus
sir.
thuringiensis species. Right? Cry1AC, protoxin, is a crystal protein produced by the bacterium Bacillus thuringiensis during sporulation. Cry1AC is one of the delta endotoxin produced by this bacterium which acts as insecticide. So what you have to remember, Cry1AC or any other forms of Cry gene, they are mainly used as an insecticide. So because of this, the genes for these have been introduced into commercially important crops by genetic engineering, like cotton, tomato, and tomato by Monsanto, in order to confer pest resistance on these plants. So once these genes are introduced into the crop, they develop a resistance to pest. So that insects cannot do any harm to these crops. Otherwise, you may be really surprised if I will say many times the crops grow very well. One fine day, you see that one insecticide starts attacking. Within no time, the entire acres together of the land or crop will be affected by these insects or pests, insect pests. Now, let us look into pest-resistant plants. Several nematodes. I hope you remember. Nematodes are nothing but which members? Nematodes. Can anyone tell me? Nematoda. Nematoda is the, yes, Pratiraj, can you tell me? Namrata, Lohit, what are the nematodes? Namrata, you know? Nematoda. Pardon? Yeah?
Roundworms, sir.
It is not a roundworm. Nematode. Now, of course, nematoda is the only one class under phylum Ascomynthus. So therefore, all Nematelminthus member, Nematelminthus is otherwise known as Ascomynthus. Ascomynthus and Nematelminthus, they are both one and the same. All Nematelminthus, since they belong to only one class, Nematoda, we call Ascomynthus members or Nematelminthus member simply nematode. Several nematodes parasitize a wide variety of plants and animals, including human beings. A nematode, Meleidogyne incognita. If at all there is a spelling mistake in your NCERT book, correct it. Meleidogyne incognita. Is it spelled like this only, or many changes there in the spelling what you have in your book? Kanisa? Blessy? Navya? Janani?
Same, sir.
Kavita? Same. Okay. The nematode Meleidogyne incognita infects the roots of tobacco plants and causes great reduction in yield, which it infects roots of tobacco plants and causes a great reduction in the yield. This is what you have. Nematode Meleidogyne incognita. They get introduced into the root and spoil the yield of this tobacco. A novel strategy was adopted to prevent this infestation, which was based on the process of RNA interference. RNA, small I. RNA interference. You'll be asked a short note on what is RNA interference. RNA interference is interfering in the formation of RNA. So a novel strategy was adopted to prevent this infestation. That means infestation or infection of this particular plant by the nematode. So nematode worm. So a novel strategy was adopted to prevent this infestation, which is based on the process of RNA interference. RNA interference takes place in all eukaryotic organisms as a method of cellular defense. So in general, we do come across what is called cellular defense. One thing that you should know, whatever that happens in our body, we say gene expression, we say gene is doing that, gene is doing this and all. The gene, in order to express, it has to, how it gets expressed? It gets expressed by the formation of a right type of protein. So ultimately, the gene acts in bringing out or in producing the correct type of protein. So what is happening here? RNA interference takes place in all eukaryotic organisms as a method of cellular defense. Now, in our body, many times our body has to defend against some disease-causing pathogens or against one or the other event that's taking place. If at all our genes work, always the genes work in such a way to defend against any of these pathogens, right type of this protein should be produced. So this is how all eukaryotic organisms, sometimes if they come to know that, yes, this protein is getting produced by this pathogen, and I should see that it is not going to take place. What it does, it interferes at the level of mRNA transcription. So RNAi takes place in all eukaryotic organisms as a method of cellular defense. So now let us see what is happening. This is what RNA, and here you have RNA interference. It stops RNA, this is a part of the DNA, it stops RNA getting transcribed. So that is known as RNA interference. I'll show you that. This method involves silencing of a specific mRNA. Now, DNA might have produced an mRNA, but mRNA, production of mRNA from DNA is known as transcription. This mRNA ultimately goes to cell cytoplasm and tries to bring the production of a or direct the production of a protein. So now this mRNA is silenced. This method involves silencing of a specific mRNA due to a complementary double-stranded RNA molecule that binds to and prevents the translation of the mRNA. So it never formation of mRNA from DNA is known as a transcription. mRNA leaving the cell nucleus and going to the cytoplasm and resulting in attraction of this relevant tRNA, bringing amino acid, pooling them together and forming the relevant protein is a translation. So what is happening? This method involves a silencing of a specific mRNA due to a complementary double-stranded RNA molecule that binds to and prevents the translation of the mRNA. Translation is prevented. The source of this complementary RNA could be from an infection by viruses having RNA genome or mobile genetic elements, transposomes that replicate via an RNA intermediate. So, how this mRNA is silenced? For this, the source of this complementary RNA, now you know, one DNA has produced mRNA, that mRNA is going to guide the synthesis or production of a particular protein. So this mRNA has to be blocked. For that, what is happening? Some complementary RNA is released there. This complementary RNA binds to this mRNA and makes it silent. The source of this complementary RNA could be from an infection by viruses. We may go for that. Having RNA genome or mobile genetic element, some transposomes that replicate via an RNA intermediate. So this is what is done. Now, a transposable element, here we got to the word transposomes, a mobile genetic element. What is meant by mobile genetic element? A mobile genetic element is a cluster, a group of the group of these nitrogen bases which move from one part to another part of the DNA. Mobile genetic element, we call them transposomes also, that replicate via an RNA intermediate. So a transposable element, TE or transposon, is a DNA sequence that can change its position within a genome. It can move from one part to another part, change its position within the genome, sometimes creating or reversing mutation and altering the cell's genetic identity and genome size. Transposition often results in duplication of the same genetic material. Sometimes you come across there's a piece and this genetic material may get duplicated. Barbara McClintock's discovery of these jumping genes earned her the Nobel Prize in 1983. So it was Barbara McClintock. She has, for the first time, she has noticed that there is what is called jumping gene. Some of the gene in the DNA, they may get cut at one end and they may get just fused with another end. So this is that is sometimes jumping, or some of the genes may get duplicated. This is what it was noticed, and these are called transportable genes, or it is called jumping genes. This is outside your NCERT, just for your information. So the introduction of DNA was such that it produced both sense and antisense RNA in the host cell. This is another aspect. What is that? So whenever we just introduce some part of this from viruses or from some other sources, the introduction of DNA was such that it produces both sense and antisense RNA in the host cell. What is this sense and antisense? You might not have heard before, and it's the first time I show you that the sense and antisense. These two RNAs being complementary to each other, that the sense and antisense, formed a double-stranded RNA that initiated RNAi and thus silenced the specific mRNA of the nematode. Now, let me just tell you what is this sense and antisense strand. Look here. This is one strand called sense strand. This is another strand called antisense strand. Sense strand and antisense strand. So whenever this sense strand, antisense strand having part of DNA is introduced into the one particular host, that creates some sort of a blockage in the mRNA, whatever that is going to transcribe. So here, I want you to remember is that If at all you are asked what is sense stand and anti-sense stand, remember sense stand, here I have given the definition, sense stand is directed from 3' to 5'. Anti-sense stand is directed from 5' to 3'. Of these two stands, which stand is a coding stand, which stand is a non-coding stand? Come on. By looking at the diagram, can you tell me which is the coding stand and which is the non-coding stand? Hello? Hello? I want every one of you to reply. You can't keep quiet. There are two stands here, sense stand and anti-sense stand. We can see the diagram very clearly. You tell me which is the coding stand, which is the non-coding stand. I have already told about this in my previous classes. Come on. Pardon?
Sense
Pardon?
stand is a not sense stand is a non-coding stand.
Sense stand is the non-coding stand. Anti-sense stand is the coding stand. Is
Coding
it
stand.
Rakesh?
Yes, sir.
Pratiraj? Whether Rakesh is correct or wrong? Harshavardhan? Lohit? Anyone? Vishwas? Sarvottam? Navya? Janani? Kanisa? Blessy? Aisha? Tanusri? Savitri? Jayasri? Kavita? Nadira? Viswita? Hello. Navya, what are you doing?
Yes, sir.
No, yes, sir. What are you doing there? Answer my question. Simply putting on this mobile and going somewhere?
No, sir. Even I'm facing network issues. The Airtel they have shut down, so I'm like sitting on my phone right now.
That is the God-given blessing to all these online students. Network issue. Very important, very beautiful word for everything. Sometimes it is real, sometimes it is a weapon to escape. What is Diti? Namrata? Which is the coding stand, which is the non-coding stand? Hello? Namrata, what are you doing?
Yes, sir.
What are you doing?
Sir, is it lagging, sir?
Which one? I
I'm
am
answering
asking.
from that time, yes.
What are you answering? I am not hearing at all. Now my question to all. Which stand is coding stand? Which stand is a non-coding stand in DNA?
Antisense stand is coding.
Pardon? Who is this?
Blessy, sir.
Pardon?
Blessy, sir.
Yeah, tell me.
Antisense stand.
Which one? Antisense stand is?
Coding.
My question is that, yeah, coding stand and non-coding stand, what is the difference between coding stand and non-coding stand? What do you mean by coding stand? What do you mean by non-coding stand? Here, in the sense stand and anti-sense stand, there are two stands, which is the coding and which is the non-coding. Which is the coding stand? Blessy? Blessy? You are right.
Antisense.
Anti-sense stand is
Coding stand.
coding stand and sense stand is Non-coding. Can you see here? What is this? Can you read? Yeah, which is the template? Which is the template?
Sense strands.
5 dash to 3 dash.
Template in this 3' to 5', 5' to 3', in this direction. 3' to 5', 5' to 3'. Which is the template strand?
Sense strands.
Sense strand? Can you see this diagram? Can you see?
Yes, sir.
Can you see?
Yes.
Which acts as a template?
Antisense.
Antisense strand.
Antisense strand is a template.
Antisense.
Yeah, antisense strand is a template strand. Is it called coding or non-coding strand?
Non-coding.
Non-coding strand. So, do you remember a sentence I have passed, statement I have passed? A coding strand is called a coding strand, but in real sense, it is not coding. Understood? Followed? Have you got your doubt cleared or still you have the doubt? So, out of this antisense strand and sense strand, antisense strand is a template strand. It is a
Non-coding strand.
non-coding strand. Okay, take down that point. Very important. Antisense strand is a template strand and it is also called non-coding strand, but it is coded. Rakesh,
Yes, sir.
is it clear?
Yes, sir. That one.
No, you told other way. You said it is coding strand. I know. Am I right?
No, sir. I said the antisense strand as a non-coding strand.
Non-coding strand. Also known as template strand. Okay, good. Right? Now, the consequence that the parasite could not survive in a transgenic host expressing specific interfering RNA. So always, whenever this DNA is introduced, it blocks the RNA that's produced, it blocks the functioning of the mRNA, and therefore it is called mRNA interference. So I repeat here, I don't know whether you understood or not. So this is about transportable element. I started from here. What is that? RNA interference. What is that? A novel strategy. Always it is asked for short note or 5 marks question. A novel strategy was adopted to prevent this infestation of the nematode, which is based on the process of RNA interference or RNAi. RNAi takes place in all eukaryotic organisms as a method of cellular defense. So this is a RNA interference. Right? This RNA, mRNA, cannot function anymore. This method involves silencing of a specific mRNA due to a complementary double-stranded RNA molecule that binds to and prevents the translation of the mRNA. So, whenever this complementary double-stranded RNA molecule is introduced, it binds to mRNA and makes it non-functional. It is not available now to do translation function. The source of this complementary RNA, as I mentioned, could be from an infection by viruses having RNA genome or mobile genetic elements, what is known as transposons that replicate via an RNA intermediate. So you need not bother much about this, but just remember that. The introduction of DNA was such that it produced both sense and antisense RNA in the host cell These two RNAs being complemented to each other formed a double stranded that initiated RNAi and thus silenced the specific mRNA of the nematode. So mRNA nematode cannot function anymore. So there is no production of protein by the nematode. So this is what I already showed you. The consequence of that, the parasite could not survive in a transgenic host expressing specific interfering RNA. Parasite could not survive in a transgenic host because in the transgenic host we have introduced this RNA, virus RNA. So this RNA binds to that of the parasitic RNA, making that parasite to die, could not survive. The transgenic plant therefore got itself protected from the parasite. Transgenic plant therefore got itself protected from the parasite. Understood? So what we are doing here, by introducing this RNA or DNA from foreign source, we are making the mRNA of the parasite stopping getting translated, right? So using agrobacterium vectors, nematode-specific genes were introduced into the host plant. You see another one. Using agrobacterium vector, nematode-specific genes were introduced in the host plant, and these nematode-specific genes will not permit the nematode to survive. It will make the nematode to die. Host plant generated double-strand RNA triggers protection against nematode infestation. Host plant generated double-strand RNA triggers protection against nematode infestation. This is A, roots of typical control plant. B, transgenic plant roots 5 days after deliberate infection of nematode but protected through novel mechanism. So the moment whenever these RNAs are introduced, that makes this parasitic RNA stopping to get translated. Now let us look into biotechnological applications in medicine. Is it understood or you want any further explanation of that? mRNA interference, RNAi, mRNA interference, RNA interference. Namrata, have you followed?
Yes, sir.
Jyoti,
Yes, sir.
Nadira, are you there? No. Vismita, are you there? Vismita. Aisha. Okay. Now coming back to biotechnological applications in medicine. Yeah, who is this?
Aisha.
Aisha, you came? Good. I wanted to check whether you could come or not. Good. Good that you come. Now let us study biotechnological applications in medicine. The recombinant DNA technology. You know what is the recombinant DNA technology? Adding a piece of DNA into the plasmid, then adding it into the host plant. The recombinant DNA technological processes have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. Many of the drugs, whatever you get, whatever you use for treatment of diseases are product of rDNA technology. Further, the recombinant therapeutics do not induce unwanted immunological responses as is common in case of similar products isolated from non-human sources. So, recombinant therapeutics. What is a therapeutic drug? Therapeutic drug is the drug used for treatment of diseases. So, the recombinant therapeutic drugs developed by recombinant DNA technology do not induce unwanted immunological response, which is very common from non-human sources, if at all you want. At present, about 30 recombinant therapeutics have been approved for human use all around the world. In India, 12 of these recombinant therapeutics are being used in the market or presently available in the market. So what is this? These recombinant therapeutics, which are the drugs which are used to treat some of these diseases, they are manufactured by rDNA technology, recombinant DNA technology, because it generates less unwanted immunological response. Now let us see genetically engineered insulin. Management of adult onset diabetes is possible by taking insulin at regular time intervals. I hope you might have heard about different types of diabetes. Who will tell me what all the different types of diabetes you know? Come on, any one of you? What different types of diabetes you know? You might have been taught in the last year in your colleges. Hello? Anyone? You tell me what diabetes you have heard about. Is there any types of diabetes?
Diabetes mellitus.
Pardon?
Diabetes mellitus.
Who is telling this? Kanisa? Navya?
Rafa.
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 Rafa, diabetes
Yes, sir.
mellitus and another one? Diabetes
Diabetes insipidus.
insipidus. That is different. That diabetes mellitus is the sugar diabetes. You know what is the diabetes insipidus is called? Otherwise, Rafa?
Type 1
Huh?
diabetes.
No,
Type 1
don't come.
diabetes.
No, that is different. Are you telling that diabetes insipidus is the type 1 diabetes? Rafa?
Yes, sir.
No,
Yes,
it
sir.
is not. It is not. Yeah, if you only want to know, diabetes mellitus and diabetes insipidus, these are the two different types of diabetes. One is sugar diabetes and another one is water diuresis. You know, in some people, there will be glucose in the urine. They pass the sugar in the urine. That is diabetes mellitus. Some people, they pass a very dilute urine, too much of urine. This is what is called water diuresis. And this is generally due to non-functioning of or non-secretion of a hormone called, can you tell me? Can you tell me, Rafa?
Insulin.
No. I am telling about diabetes insipidus. You are right about diabetes mellitus is due to less release of insulin. I agree. This is different. Genuine.
Glucagon.
Huh?
Glucagon.
Pardon? What is that hormone?
Glucagon.
No. You have another diabetes, what is called water diuresis, diabetes insipidus, and this diabetes insipidus is due to less secretion of anti-diuretic hormone. What is it otherwise called? What is it?
Vasopressin.
Vasopressin. So that is different. I am asking you, among this sugar diabetes, you have different types of diabetes. Rafa, now what you said is correct. What you said? You said one something, you know? Rafa. Why are you sitting quiet now? Rafa?
Yes, sir.
What is that? Type 1 diabetes. Type 1 diabetes. Type 2 diabetes. You have two more. You know. Gestational diabetes. You know. Gestation diabetes.
No, sir.
Stress-induced diabetes. These are all different types of diabetes. Type 1 diabetes is also called, are you taking down or simply sitting? Prithvi, you are not taking down. Am I right?
No, sir, taking down.
Huh?
Taking down, sir.
Okay. Type 1 diabetes is also called, what did I say? Juvenile diabetes. This juvenile diabetes, nowadays, it is appearing in such aged youngsters. Even there are reports that juvenile diabetes has become very common now at your age. And also to the surprise, sometimes this juvenile diabetes is found to get reported in the kids of 6 to 7 years. What is that? Juvenile diabetes. Type 1 diabetes. Type 1 diabetes, what is the type 1 diabetes? In type 1 diabetes, insulin is never produced. No. Absolutely no production of insulin. As against this, there is another diabetes, what is known as a type 2 diabetes. Adult onset diabetes. Type 2 diabetes. In these individuals, insulin is a producer, but receptor cells, they do not respond to this. When the receptor cells, if the receptionist does not welcome, what is the use of the guest? Guest has to go. Whenever this hormone comes, the receptor cells are not diagnosing it. That is the type 2 diabetes. Type 2 diabetes is seen generally in the adult. And type 1 diabetes, do you know? Let me tell you a case history. In the type 1 diabetes, if at all anyone develops a type 1 diabetes, what will happen? Come on. Anyone has the idea? Have you heard about this? Type 1 diabetes individual? Harshavardhan, Sarvottam, Siddesh, Charan. Who else? Sarvottam, Siddesh, Charan, Harshavardhan, Vishwas. Have you heard about this? Type 1 diabetes individuals, if they want to survive, they have to take insulin injection throughout their life. Have you heard about this? Rafa, Aisha,
Yes, sir.
Blessy, have you heard about this?
Yes, sir.
Yes?
Yes, sir.
Then Rafa said, no. Rafa,
Yes, sir.
you said no, you know.
Sir, I
Did
knew
I say
about
you?
this, but I didn't know it was type 1 diabetes.
Okay. You know what is that? How many times you have to take? In the morning or at night? Rafa? Aisha?
Before food, I guess.
Not you guess. Star of the day. Aisha? Good that you have come to the class. Okay? Before food. Aisha, then how many times?
Sir, twice.
Hmm?
2 times.
Only 2 times? You
3.
take, but only 2 times? Before breakfast, before lunch, before dinner. Is it a swallowing? Is it a swallowing insulin tablet?
No, sir. No, sir. They inject it.
Injection. They have to take injection. Who has to give injection? Who has to give injection? Who has to give injection? Who has to give injection?
Family member?
No. Who will come, Aisha? Who will be having that much of time to give the injection? He or she should get trained to prick themselves. If you see such individual who are suffering from type 1 diabetes, if you see their body, They prick themselves to the region which is generally hiding, kept hiding. They go to washroom, prick that to their buttock or to some other place, and they come. When they have to prick, when they have to take the prick, at any time, when they have to get the prick, at any time,
Before
no,
food.
just before the food. I should tell me if at all they take the injection about 1 hour before. Now let us say that there's a guy or there's a dame, she is suffering from type 1 diabetes. She took the injection, she has gone to some other place, thought that she will be serving lunch now, and she has to take injection before lunch, and she saw that that noise is coming up, a movement of the plate and keeping the plate and all. She goes to washroom, she pricks herself and comes back, but food is not served for another 1.5 hours. What will happen? What will happen? Huh? Huh? Huh?
Insulin level will increase.
Huh? She will get into coma. She will fall unconscious. The moment insulin injection is taken, within half an hour, she should get the glucose. Glucose level comes down, she falls. She falls unconscious. Therefore, it is very difficult. And she cannot go outside. She has to always with the insulin injection in her hand. She should learn to prick herself. And not only that much, I shall tell you one of the case, history. One particular girl student got this type 1 diabetes. After some time, after the completion of her engineering course and all, she got married to a person with whom she has told everything. After that, she became pregnant. You know, now, through to the pregnancy period, she has to see that the insulin level is monitored perfectly, the glucose level is monitored perfectly. That means for every 2 hours of interval, she has to take a blood drop. She has to measure the glucose level. If the glucose is less, she has to eat something glucose. If the glucose level is high, she has to monitor. Every 2 hours interval, daytime it is okay, nighttime 6, 8, 10, 12, 2, 4, not 1 day, not 2 days. She has to do that throughout her gestation period. Why that much of strict monitoring of this? If at all the glucose level falls down, it would affect the fetus. It would affect the fetus development. It's very easy, but very difficult to manage. This is what the type 1 diabetes. Therefore, but the question is that why that present day youngsters suffer from this diabetes? You know what is another one? Another incident? Which one is at high level? Increased level amongst youngsters? You know? It is hypertension. Nowadays, good number of guys at the age of 20 plus are seen to suffer from high blood pressure. Why it is happening? Why that present day generation, present day youths are suffering from type 1 diabetes, even this hypertension, and many times even heart attack, heart fail. You know what are the reasons for this? What are the reasons for this? Namya, what are the reasons for this? Aisha, Rafa, what are the reasons for this? The reason for this is said as present day lifestyle. Present day lifestyle. It is unfortunate. in one of the institution where I had gone to give lecture, I came to know that one girl student was perfectly all right, had come to write SSS exam. She wrote the SSS exam. After finishing the paper, when she came out, she all of a sudden fell down. Fell down. And immediately it happened at Sringeri. Immediately principal called me asking, sir, do you have anyone? Doctors at Bangalore, so that we can refer that particular student to Bangalore. Immediately that particular student was brought here and she got into some hospital. Immediately that hospital people said it has to be referred to NEEMANS. She was taken to NEEMANS and she collapsed there and she died. And the reason was cause of death. It was not known properly. But she suffered from heart attack at the age of 16. Therefore, my request is you. What is it? What is my request? What is my request? What I am going to tell you? Come on. Aisha, Rafa, what I am going to tell you? Diti, what I am going to tell you? Namrata, what I am going to tell you?
To have proper lifestyle, sir.
To have proper lifestyle in the sense, at least have minimum 15 minutes of exercise. Would you? Would you? Would you? Would you? Would you? No
Yes,
voice.
sir.
No voice. No voice. See, certain things
Yes,
should
sir.
happen. Once it happens, you cannot correct it, repair it. Good that she died. If at all she has survived, if at all she had survived, she had to suffer from paralysis. Some part of the body not working. We don't know why it is happening. Maybe that 1 in lakh, 1 in 10 lakh, like that. If at all it happens tomorrow to us or to our family, then it is difficult. So, type 1 diabetes, type 2 diabetes. Type 2 diabetes is elder diabetes, they have to take tablet and they have to manage. There is another diabetes called stress-induced diabetes. Have you heard about that? Have you heard about stress-induced diabetes? Stress-induced diabetes will appear, disappear. Most of the principals of most of the colleges, they do have diabetes. They do have diabetes. Why? Why? Because they get always stressed because of students. Because of students. So they have stress-induced diabetes. A person suffering from stress-induced diabetes will develop diabetes. He has to take or she has to take tablet. Sometimes it comes down, again it develops, and they will live with the diabetes throughout. One more type of diabetes is the diabetes that is developed during pregnancy. What is it called? Gestational diabetes. Gestational diabetes. Diabetes developed during pregnancy. Once the woman gives birth to, pregnant woman gives birth to baby, that the diabetes vanishes. But, what is that? But that lady getting again diabetes is there. There is a possibility. She is prone to diabetes. She is prone to diabetes. She may get diabetes at any time in her life if at all she neglects her lifestyle. One more diabetes is medication diabetes. Taking some tablets to treat some disease makes the person to get the diabetes. If at all one takes some tablet for one particular problem, health problem, without his knowledge, he develops a diabetes. As long as he is taking the tablet, he has to take this tablet also for diabetes. Best thing for all this is prevention is better than cure. So what we have to do? We have to be healthy. To be healthy, what we have to do? We have to go for little bit exercise every day. At the same time, we must be very much careful our food habits. Food habits. It is always not good to eat too much of ice creams, too much of sweets. No, you may like them, you may like it very much, but you should have the control. Right? Now, I hope, now let us come to genetically engineered insulin. Management of adult onset diabetes is possible by taking insulin at regular time intervals. Insulin used for diabetes was earlier extracted from pancreas of a slaughtered cattle and pig. You may say that you are strictly vegetarian, but the insulin that is used for the treatment of diabetes was earlier extracted from pancreas of a slaughtered, killed cattle and pigs. Insulin from an animal source caused some patients to develop allergy or other types of reaction to the foreign protein. Some people, the moment it is given injected to their body, they started developing allergy or some other health complications. Insulin consists of two short polypeptide chain. You must know about that. Chain A and chain B. There are two polypeptide chain, and these chain A and B are linked together by disulfide bridges. Shall we see? So you have here chain A and chain B. This is the chain A, and this is with the 21 amino acid. Chain A, 21 amino acids only. This is what the different amino acids we have, you know. And this is the chain B. And this chain B has a 30 amino acids. And these are the disulfide bridges that keeps the two chains connected to each other. Right? This is human insulin. Right? Chain A, sometimes it is asked, how many amino acids are there in chain A? How many amino acids are there in chain B? Chain A has 21 amino acids, chain B has 30 amino acids. Okay, clear? So this is how the figure showing maturation proinsulin into insulin after removal of C peptide. Okay, so this is what you have. This is the C peptide. You have one piece and this another piece, and you do come across in between. This is what proinsulin. And once this C peptide is freed, it becomes proper insulin. So this is what you have. Which one is A chain and which one B chain here? Green one is B chain, blue one is A chain. Am I right? Am I right? Hello? Hello? Which is A chain, which is B chain here? Hello? You can't make out? You can't make out which is A chain, which is B chain here? Can you make out? Harshvardhan? Can you make out? Hello? Why you people are keeping quiet? Whenever I ask you a question, you should respond to that. Namia?
Yes,
Hello?
sir. Something to
What is this chain? Green color. You can't make out which is B chain and which is A chain. Navya? Hello? Namrata? What did I say before? A chain has how many amino acids?
21.
B chain has?
30.
30.
So now which is A chain and which is B chain?
Green one is
green
HA.
is A.
Green is?
B.
Blue is?
Blue is B.
Green is B. Huh?
B.
Green is B. Blue is A. Am I right? Is this blue A? Which is shorter and which is shorter, longer here?
Green is shorter, sir.
So green is what?
A.
A.
Green is short, A. B is blue. In mammals, including humans, insulin is synthesized as a prohormone, like a proenzyme. The prohormone also needs to be processed before it becomes a fully mature and functional hormone, which contains an extra stage called C-peptide. So in between A and B, we come across a C-peptide. When C-peptide is there, it is called proenzyme, prohormone. This C-peptide is not present in mature insulin and is removed during maturation into insulin. The main challenge for production insulin using recombination DNA technique was getting insulin assembled into a mature form. I hope you understand this. In 1983, Lilly, an American company, prepared 2 DNA sequences corresponding to A and B chains of human insulin, and it's called human insulin, and introduced them in plasmids of E. coli to produce insulin chains. Chain A and B were produced separately, extracted, and combined by creating death sulfide bond to form human insulin. It is called humulin, right? You must remember, it is Lilly company that have prepared this. Okay, I shall stop it here. We shall continue with the gene therapy in tomorrow's class. Okay, clear? And I may start the new chapter tomorrow.